So I am going to tell you, a story of myself, kind of from the beginning to the today. So I hope you will enjoy the talk. And the title of the talk is, Nanobiophotonics and Nanomedicine. I'm sure you all are familiar with this terminology, but I will try to explain what I mean with these terms as I speak. So here's the outline of my talk. And this is basically, first, I am going to explain to you what I mean with nanobiophotonics. Actually, I'm referring to surface and Raman scattering. Some people call it surface and Raman spectroscopy. It's both are fine. Then the applications, some selected applications for these challenging problems that humankind face. And I include a few slides about microbial identification and cancer diagnosis. Then I'm going to move to nanotechnology idea in medicine. Basically, I'm referring again here to nanomedicine. And of course, not only the idea and the materials are used, these nanomaterials used in medicine. Then I'm going to give you some examples of these materials used in my research. These are boron nitride-based nanomaterials and DNA nanotechnology. And again, the surface chemistry versus toxicity of nanomaterials. And I'll conclude my talk. Well, I mean, I don't remember when, but from the beginning of my career, I kept showing this slide to before my every talk. And this is the idea is when I returned to Turkey, and I knew nanobiotechnology idea, and I'll try to explain to you in a few minutes. And then also I knew that nanotechnology may not be the solution for every problem that we face. So still we need to stay on the molecular side. And I named my research group as Nanobiotechnology and Molecular Engineering Group. So idea was to use these nanomaterials and molecules, build structures and make them and use them to solve problems that we face. For example, in nanomedicine. Of course, if you are talking about medicine and using these materials in medicine, we need to know the toxicity of these materials, right? So what causing the toxicity to the living systems? It's a very complex story, and I wish I had time to tell you more. And then also to use these structures and surfaces and these nanomaterials, and for other applications like making intelligent materials, for example, or surfaces for sensing, and also getting information from living systems, including microorganisms and living cells and such. Then the last one was my hobby, the self-assembly of nanomaterials. And of course, then it refers to DNA nanotechnology because it is considered a molecular self-assembly. So as I told you, it's nanobiophotonicks. What is it? It's just not a hot research keyword. As actually, it refers to collect information from nanometer resolution using nanostructures and using light. And if you are using these structures for biological applications, of course, it becomes nanobiophotonicks. So basically, I am referring again to surface and Raman scattering here. So I will tell you just a little bit before I move on to the surface and Raman scattering. I wish I had more time to tell you about the Raman scattering and its background, molecular background, but we call simply it's an inelastic scattering. As you see on the at the bottom of the slide here, this is a complete Raman spectrum from carbon tetrachloride. As you see, it is not just one piece, one spectrum as we see on the instrument that we use. And we see Stokes and anti-Stokes lines. Both are used in research and technology, but the Raman scattering is strongly depends on the polarizability of a molecule, which is the electron oscillation, electronic structure. So then I think this is enough for now. Let's move to surface and Raman scattering. Well, I mean, in early 1970s, it was discovered that when a molecular structure is brought to the surface of a noble metal, it was, at that time, it was a silver electrode, the Raman scattering enhanced tremendously, up to 10 to 7. So it was not very clear at that time what causing this. And there are two groups, one from one England and from one United States. They explained this with chemical enhancement and electromagnetic enhancement. But actually we know what is happening now. It's basically these two theories are still holding okay. But we know that the electron system of a molecule couples with the electron system of the noble metal. Actually, we know now that not only noble metals can enhance Raman scattering, any system, even polymeric structures with loosely oscillating electron system can enhance Raman scattering. Actually, if the molecule's electron system couples very nicely to the whatever is the substrate they are on, they can actually, their electron system can be, oscillation of the electron system can be enhanced. Then this is the basis of the Raman scattering, because photons, as they collide with the electron system of the molecule or molecular structure, so they bounce back, either losing or gaining energy or not, changing the energy. So then this is is the process basically then at the moment that I can explain. So here you see this, the plasmonic structure at the right-hand side, at the right corner. So these are, for example, a reciprocal particle, and deep, because some people attempted to explain the surface enhancement using surface plasmons, and where they localize. Of course, this surface plasmons or the extension of the electron system of the metal. So that is exactly the same thing that we are talking about, the same thing, surface plasmons or the extension of the electron system as they oscillate, okay? And let's move to the next slide. So in a source experiment, the experimenter should be careful with many things. The substrate they're using, laser wavelength, molecular structure, and so forth. And actually, it might be a very misleading, the person who is doing the experiment can get very wrong results and maybe very misleading, reach very misleading conclusions. But I think, if you try, when you start working on in laboratory and you realize all these things are important. And the question here, what kind of substrate you should use? There are people use surfaces and colloidal particles. And surfaces are made through many different techniques, including lithography. And this could be, soft lithography or E-beam lithography and so forth. The enhancement factor is in the range of 10 to 5 to 7. And but the making colloidal nanoparticles are easy. Of course, still you need to be experienced making this. And but there are issues with that. I mean, it's not easy to control the reproducibility of from batch to batch, and but the enhancement can go up to 10 to 12 to 10 to 14, which tells us that this technique can be used even for single molecule detection. So in my research, we always used silver or gold nanoparticles, okay? And we can synthesize this easily, and we've been doing this for years. And if you're using this nanoparticles in your research, you need to be careful with the size issue here, okay? And as you see here, I told you earlier, I am applying this technique in biology. So in complex biological systems, if you look at the size of the structures here, proteins or molecules and viruses and yeast and in eukaryotic cells, and the noble metal particles here that we use in the range of this 5 to 200 nanometers, maybe larger. So you need to understand the size relationship, because small molecules stick to nanoparticles, but the large, if you, for example, say yeast, the particles stick to this yeast cells. And sometimes if we are using eukaryotic cells, particles enter into the cells through endocytosis, or depending on the size, it could be different routes. So here are the list of the applications that we use over the years. So tissue differentiation for, we are referring to cancer here, and cell death monitoring, nanotoxicity assessment from the eukaryotic cells, of course, and also the cancer diagnosis from eukaryotic cells and from blood, which is called liquid biopsy, and the identification of microorganisms, and the understanding the discrimination of biofilms and so forth. It has been, a journey of, extraordinary journey over the years. We accumulated, there's so much that I cannot include here. And, but the key point here is the sample preparation, because the idea is to bring the molecule or molecular structure that you are interested in working together in a nice packing, these nanoparticles and the molecular structures together. So this is, we used many different approaches in the past. Spotting and drying, don't think it is a very simple process. It is really complicated. And I hope you know the coffee phenomenon. And actually we contribute to literature through this. And when we realized that, we thought we discovered something, but we realized later that we did not really discover anything, because protein, proteomics people all the time use this suspending droplets and drying, suspending and drying. But anyhow, and we applied this approach in our research, and we show that it is good. Anyway, we use convective assembly, and even we dipped a hydrophobic tip into a drying droplet to control the distribution of molecular structures and nanoparticles in the drying droplet area. And we have, I think we showed the world that, this is an important issue in this kind of research. So I'm going to tell you just a brief story of microorganisms, how we utilized this technique for bacterial identification, also the yeast and viruses. We never published with viruses because I was scared with the results that, it was confusing. And we did not have the facility to work with viruses, but we have a lot of work with bacteria and yeast. So then, where is the cursor here? Yeah, actually this is the spectra of a gram-negative bacteria. And then we know to some degree, what are these bands, where they come from. And the idea, we know that this actually originates from the molecular structures on the surface of bacteria, or whatever is the cell, and some metabolites, they contribute to overall spectra. Maybe this is enough to tell you, what we see on a spectrum. And so I'm going to tell you a story that we attempted to use this technique in clinical settings. Actually, it was an attempt for translational research. So, let me tell you something, about the translational research. Translational research, is the research that your fundamental dreams end and the reality kicks in. So because the real world is quite different from what you imagine in your fundamental world, because that is actually the comfort zone that you are happy there. You just keep playing with certain things in your laboratory and whatever you have. But when you move into real world, it's a different story. So, and if you look at urinary tract infection causing microorganisms, and you see this slide is showing that. And of course, there are many microorganisms are possible. And so what happens in a real setting, if you go to hospital and if you have a burning sensation in your urinary system, and they actually collect your urine, and immediately they tell you if you have a infection or not. But the criterion to say that if the number of microorganisms is more than 10 to 5, CFU in per milliliter, they tell you, oh, you are infected. Otherwise, you're not. You just go home and drink more water and this and that. But if they decide you're infected, they need to figure out what microorganism is causing this. Okay, then the second step comes, and it takes up to from 24 hours to 72 hours, depending on the type of microorganism they can grow on culture medium. When I talk to clinicians, they told me that maybe my technique can help them to identify, the microorganism fast in a shorter time, less than 24 hours, for example. So then what we started with this idea, and we developed a sample preparation method. Then we used our technique, as you see here. And I'm not showing the complete data here, but see the E. coli, and all these microorganisms are on the PC plot. And as you see here, this is the, again, another plot. But I mean, what I'm trying to tell you here is that it is possible to identify a microorganism in 1 hour, but you need to culture it. You need to put it on a culture plate and start culturing, and 1 hour later, you collect the microorganism, all these, the microorganisms are starting to form on the plate, and wash them and take the spectrum in 1 hour, approximately 1 hour, you can identify the microorganism. This was something, and it was something that, I mean, I had a dream to use this technique in clinical research, and this is actually showing you by how spectral changes evolve from 24 hours to 1 hour. So, and that actually clearly tells us that this technique is capable of identifying microorganisms growing on culture medium in as early as 1 hour. Okay, so now I'm going to switch to another challenging issue: cancer diagnosis. Okay, I'm sure you all know, I think, as the number of patients, people suffering of cancer increases in societies. And the definition of the cancer is abnormal cell growth. And so tumor can be benign, malignant, or fast growing, or slow growing. And this could be, the cyst instead of, being a tumor. And actually, how complicated is the diagnosis of this. And actually, there is an article I wrote in 2015, how Raman spectroscopy can contribute to this. And in that article, I studied how clinicians actually, roughly, this is not a, of course, complete picture, but how they decide if a person is cancer, on what kind of cancer, and what's the level of, the grading level, and this and that. It's actually quite complicated. So then is it possible to use my technique to help clinicians to diagnose cancer in a more reliable way, maybe faster? And because I'm not saying a complete diagnosis, because I know, and you all know, it's not that simple process. And so it may help to increase reliability, speed. Maybe we can lower the cost, and maybe we can do it earlier. So that, and the idea is there are many points in the decision process that research can contribute. I'm going to show you the first example, tissue. And this is our very early study, very interesting. And we had a brain surgeon and at the Kartal Araştırma Enstitüsü hospital. And then he came to me, and for a different reason. And I realized that he can give me the samples. And I asked him how many patients actually they see and do the surgery of daily or monthly. He said, you know, it's a quite large number. We started collecting samples because I asked him how he does this surgery. He said, you know, first we remove the tumor, then we keep cleaning the surrounding tissue because we don't want to leave any cells, tumor cells around the, you know, the tumor formation. So I asked them to label all these because it's not easy, of course, it's just a label. You know, first tumor, then the peripheral tissue and the healthy. And at the end, we didn't know if it is healthy or not. The last piece of tissue cut from the real tissue is labeled as healthy. Then we used, you know, of course, clinical diagnosis parameters to decide what kind of level of the grade of the tumor and the progress or rate and this and that. As you see on the right-hand side, right here, then there's a staining results, probably. I don't have the complete data here either. You can look at the publication. But we realized something. When we did this, we did a number of different types of cancer types. And we realized that there are two peaks. If you take the ratio, as you see here, in the tumor tissue, this ratio of these two peaks, 723 to 655, and actually it's always high. And actually this ratio increases from the healthy to tumor tissue. So which means that this technique can tell us, you know, even during the surgery, the surgeon can decide what to do, you know, where to stop. For example, during the surgery, if it reaches the healthy tissue in just 10 minutes though, you know, from the sample preparation to getting the spectra and looking the ratio of this. Of course, an algorithm can be prepared for this and it make things much faster, but, you know, we never, you know, went that direction because we need real clinicians to work with. So not easy thing again, translational stops there. We showed something, but how about the early cancer detection or liquid detection from liquid biopsy? I'm sure you know that it's a very hot topic nowadays, using body fluids to diagnose cancer and even early. So this early cancer detection is a really complicated issue. And there are so much complications. And I was at this center at Oregon Health and Science University, Early Cancer Detection Center, and they look every aspect of it, from epigenetics to you know, the circulating tumor cells, circulating RNAs, DNAs, and everything. So I had an idea because, you know, in one of my students who studied the blood and tried to not early detect, but the diagnose cancer from blood using this surface and Raman scattering. And actually he showed that this is possible. And I first did not really believe this because I know blood is, you know, blood is a mess. I mean, it's a debris. It's, you know, thrown by body. You know, imagine a river, everything is thrown into. So not easy to identify what is in there and what is what. And people are working on this so hard, and every time they get a conclusion which is fuzzy. So, but anyhow, then I was trying to understand what is causing the difference. Cancer, you know, the spectrum from a cancer patient's blood to control healthy. Of course, we think they're healthy. And there is a variation, and that's what I was trying to understand. And I think I came to a point, I have an idea what is causing this, and I hope I will have the time and the energy to pursue this. And also the other issue, the single cell analysis. Well, I mean, this is another thing that, you know, we know that even cancer or many diseases is, you know, originating from a single cell. If something goes wrong, and molecular biologists know this very well. And, but molecular biology is always time consuming. It is laborious, I mean, it is expensive. And the idea was to use spectroscopy and other techniques, novel techniques and tools to get information from living cells. So it was my idea to use my technique to do this, and how, that is, I'm going to briefly show you this. What we did was, we introduced gold nanoparticles into living cells, and we collected spectra from these living cells, but it's not that simple. We need to understand how these nanoparticles are entering into living cells and what they're doing in the cell. So because we know that, I told you earlier, I'm referring to nanobiophotonicks. As you see here on the left corner, the bottom corner, see these are the aggregates of silver nanoparticles in possibly in endolysozymes, and cell is trying to handle it, and because it's just a metal. So because the most probably, I mean, not most probably, it's definitely, some of the spectral features are originating from there. So we had to understand step by step, what is happening to these particles as they enter the cell, and to relate the cell's progress with any abnormality or whatever is happening in the cell to the spectral pattern that we observe. You can take a look at this article published in recently in Untitled Chemistry. It's a perspective article, and I think I'm sure that you will find it interesting how things are from my perspective, or our perspective, not from a molecular biologist, but from somebody who lost his identity in research. So this is the TM images, for example, this silver, the gold nanoparticle aggregates in the cells. And as I told you, because we collect information, spectra is originating from these aggregates, whatever is on the top of these nanoparticles, and these are nano size. So then, of course, it's nanostructure. Then we use light to collect this molecular information. It's nanobiophotonicks. So, and it is a lot of work, and some of my students really did a good job, and I need to acknowledge their work here. So I'm gonna switch the gears here a little bit. I don't know how much time I left. Still I have some. So I'm gonna change the gears to nanomedicine. Well, I think, I don't know how these concepts evolve into this point. And as soon as nanotechnology and nanobiotechnology idea is appeared among the scientists and researchers, of course, nanomedicine was there, because we need to use this idea in medicine. Okay, so then it's out of control. I mean, it's not even possible to classify them into something. From delivery to control release, diagnosis and treatment, biocompatible surfaces, scaffolds for tissue engineering, implants, detection and diagnosis, and so forth. It keeps going. So there are many, many, many, many different applications. But of course, there are issues too. What I'm going to show you today is that there are one material that I'm very interested in using in medicine. And I tell you why I am interested in this. Because these boron nitride based materials, and they're considered as analogs of carbon based nanomaterials. There are two types that I'm interested in: boron nitride nanotubes and the hexagonal boron nitrides. There are different types of these boron nitrides. There are different crystal structures and so forth, but the most interesting one is hexagonal boron nitride. So you may ask me why I am interested in this. It's because I'm a chemist and I know a little bit physics. So then we talked about earlier, remember, the electron structures and how electrons are oscillating, this and that. So if you look at the structure of this material, boron and nitrogen, and the electron distribution in the bond between boron and nitrogen is not equal as in the case of carbon-based materials, because carbon and carbon, their electronegativity is the same. So then the bond between boron and nitrogen is a little bit more, relatively more ionic, okay? So which means that this material can dissolve in biological medium, That was the idea. I mean, which means this could be less toxic because, you know, metal particles and carbon-based materials, they're like a stone. I mean, they stay there. If they are not eliminated from tissue or, you know, from through an organ or somewhere, and then they are there, and it may generate a lot of different problems in the biological system. And it's not a good idea to use them in medicine, though. So then, if we have the if we can utilize this material and with these properties, if we understand, you know, what might be happening in biological medium, maybe we can use these materials in medicine safely, maybe for delivery and you know, targeting and this and that. But we discovered something else on the way. Okay, that became more interesting as we move on. So, and here's the applications. Usually people, what's going on? Can I see that? Okay. So, and these materials are used in making hard materials, like, you know, in polymers. And the funny thing is that hexagonal boron nitrides, they are used in cosmetics. Women use this, you know, they put their faces because it's transparent. You know, when you mix, I don't know how they do these formulations, but your skin looks brighter, and you know, it removes the, actually it's an illusion effect. Your skin looks smoother, smooth, so it's nice, and it is non-toxic, and it is used for many, even in pharmacology, to make some formulations, I think, as a filling material. But anyway, in this slide, you see these possible applications. And also we explore the possibility of modifying these particles, these materials. How? We can modify through the nitrogen atoms and the structure, or boron atoms. Because we know that, I'll show you the pictures, they are not very beautiful material, just like any other nanomaterials, the mess. So they have defects and, you know, the edges. So they are free atoms. Then we can, you know, make them either ammonia amine groups or hydroxyl groups for further modification. That's a good thing too. So then are they toxic? And we know that. I mean, for we modified boron nitride nanotubes and actually introduced hydroxyl groups through boron atoms. So it is, as you see, you know, they are not toxic. Actually, we are not the only one who's claiming that this material is non-toxic. Of course, the dose is important here. You know, if you put too much, and also their hydrophobicity could introduce some tricks, because it's not easy to disperse this material in culture medium. So, and also they may interact with this hydrophobic and toxic drugs. Any drugs that we tried, doxorubicin, which is commonly used in cancer therapy, and this can be a good thing to carry drugs using these structures. And actually, a publication we show that we compare these hexagonal boron nitrides and the boron nitride nanotubes, which one is better, and we think that these molecules also can enter into the, you know, the tube and also stick to the surface, and it is good candidate for such applications. And the other good thing is that actually here these two images are showing that how they make into the cells and they stay in the cytosol. And because these tubes are labeled with doxorubicin, because doxorubicin sticks to boron nitride nanotubes, and they don't, you know, they stay with the tubes, and maybe they are diffused into cytosol, because also doxorubicin is a fluorescing molecule, so it was easy for us to visualize that what is happening when they make into the cells. So the other application is that you use these materials because they have a very high tensile strength, and we edit just a little bit of this 0.5% volume to mass into the, for example, this gelatin scaffolds, because gelatin scaffolds are very good scaffolds, but they're terrible. They have a terrible tensile strength. So they just collapse, and cells cannot adhere themselves and they become useless. When we added this material, it was fantastic. The cells were adhering very nicely and then they survive longer. And I know why these materials made these scaffolds better, because cells, life does not like either hydrophobic or hydrophilic. It goes in the Gaussian, in the middle of the Gaussian curve. It likes both, hydrophobic and hydrophilic. So because gelatin is mostly hydrophilic. So when we introduce this hydrophobic materials, very small amounts, if we disperse it very well, so then it adheres, the cells can adhere themselves to the scaffolds nicer, and they multiply faster. The other, the study was actually there was one report. They said that hexagonal boron nitride nanoparticles are good for prostate cancer. Well, I mean, because we were also interested in this, and they speculated that they could not really explain why these materials are good for prostate cancer. But of course, one straightforward idea to think that these materials are dissolving in biological medium, and it's the products, the decomposition products are doing something to this cancer cells or tumor. So they also included animal experiments. We just wanted to deep into this idea, and actually it is indeed, decomposition products are making the difference. I mean, not the particle, because by time it's very slowly degrading in biological medium. So, and the degradation products are inhibiting the cancer. And now it needs further investigation, because we do not really know the mechanism. The other was the study was it was actually promoting the wound healing. And unfortunately, I'm not going to tell you too much about this, because I think I'm going to finish my talk. But I like to just say a few things about the DNA nanotechnology, DNA and DNA nanotechnology, how we utilize this idea to make things. I'm sure I'm familiar with this, the idea. DNA is not just a genetic material. It can be used as a material too, utilizing this the hybridization properties. And the past people showed that they can make a lot of different structures using this and that. And also we also show that we can utilize the DNA hybridization to make things. As you see here, there's a single wall carbon nanotubes in the ring forms. And we believe we use this DNA hybridization. And of course, I mean, as I told you earlier, and it was our comfort zone, and not really, but we enjoyed making these rings, 50 nanometer diameter rings, but we don't know how to use them or what to do with them. We just made them and it looks good. So I mean, I hope that I'm trying to make a point here. So, but I mean, people ask you real things. Well, this is real, but I don't know, maybe 200 years later, somebody will use it for something, I don't know. But, and the idea was to use this DNA origami structure, as you see here, for drug delivery and gene silencing. We can construct this, as you see, and we just need the pieces. You need to design this. And actually there are softwares, you can design a lot of different things, but we stuck with this design, the tile, and we use this for both drug delivery and gene silencing. And actually one of my students, she showed that it is possible to downregulate 4 genes at the same time, simultaneously. And we used a structure like this. The idea was this, we put this, here's the red ones, here is the antisense morpholinos, and they target the gene where is expressed. I mean, the region of the gene which is expressed. And these are the gold nanoparticles. It helps this structure get into cell faster and nicer. And the rest is just the, the DNA. So let's see what happens. When they make into the cells, the enzyme degrade them, gold nanoparticles, and they eliminated. See this morpholino red one goes and sticks to the DNA mRNA target region, but then the translation is blocked. So that was the idea, and it worked. And then not much time here to explain all these things, but it's nice to work. And it was possible to as I said, down-regulate up to 4 genes. And so finally, I will just tell you briefly about how surface chemistry is affecting the toxicity of nanomaterials. Of course, we know there are many different parameters here: size, shape, type of material, and this and that. But we showed in the past that surface, what is on the surface is really important for toxicity, because the interface between the living system and the material. Then how they behave in the biological system is important. In the past, we modified particles, gold, silvers, and zinc oxides, boron nitrate nanotubes, boron nitrides, and this and that, with many different molecular structures, mainly biomicromolecules, because the idea was to make them less toxic, but you don't really know if you are going to make them less toxic or more toxic. So that's what we learned. And actually, this is our very early study, interesting one. We modified this with lactose. And we said the idea was to use these silver nanoparticles for photothermal therapy to kill the cancer cells. We compare which healthy cells to cancer cells, which one is uptaking these lactose modified ones. And we found out that the cancer cell uptaking them very fast because they have higher metabolism. And idea was to use a laser, shine a laser, heat it and kill it. But we learned that it just dies. And actually it's not non-toxic, it is toxic for cancer cells. Of course, it is doing something to healthy cells too, but comparatively, it was more toxic for cancer cells. It was interesting. But anyway, in conclusion, then I hope you're still with me. I know, and we learned a lot of things on the way. And these are the conclusions of this talk. But my conclusion, I have not concluded yet in science. And now I am at the translational stage, because now we were happy to prove our ideas, were working, and we got money from funding agencies for this. We played, and we had experience. Now we need to understand if we can use these ideas in real life, and we need to get out of our comfort zone. I'm not saying that you should give up fundamental research. It is there, and it has to be. And to move a little bit, stick your head out of the window, or we say in Turkish, put your hand under the stone and try to learn more. Still, continuous research and development is not an easy task, and no one should take it. No one should, take it lightly, and it is real, and it is really, really important for the development of societies. And I'm sure we all know that from with this last, for 1 year, remember, the pandemic, how everything screwed up around the world. And this is an old picture, of course. I had a large group, and the funding comes from the European Commission and TÜBİTAK. And I appreciate, of course, every person who contributed this work. And I like this cartoon, these two cartoon to share with after my or before my talks. And as you see, well, you know, what boss says is important. That's not how it works. So then also you need a nice project, a nanotechnology stem cell for fighting terrorists. Maybe can help you to get more money and funding, and your research keeps going. Thank you very much for your time. Where I am. Am I here?
Thank you very much for your impressive presentation. If there are any questions or comments, please feel free. Do you have any question?
Well, I think there's one, somebody said something.
I have a question.
Sure.
First of all, Çulha, thank you for inviting me to this very nice talk. It was very interesting.
Okay. Thank you very much for your attention, and we will wait everyone the next Nano Open webinar. Okay, see you soon.
What happened?
There's one question.
Do you have any question?
Asking question. Hold on.
Sorry, I'm sorry, I cannot hear. Ah, sorry.
It's okay.
Okay.
Hello, everybody. My name is Katerina Pistiki. I'm from the IPHT, working here in Jena, Germany.
Oh,
and
hey, Katerina.
This very nice talk, and it was really, really interesting. So,
thank you.
My question is the following. First of all, let me say a few things about my background. I'm a biologist, and I have worked many years in clinical research and also in routine diagnostics. So I'm actually from the other side of the tunnel, and your comfort zone is uncomfortable for me, and I think it's vice versa. So because I have the application in mind and not the way and the path you need to go until it's applicable in routine diagnostic and in clinical practice.
Yes.
So I'm working with Raman spectroscopy of bacteria. And what we try to do is to detect resistances. We use Staphylococcus aureus as a model right now, but also in general with gram-negative and other antibiotic resistances of bacteria. So going through the literature, I've seen a lot of SIRS application in this field, not only resistances, but also detection of bacteria by using streptavidin and biotin labeling.
Yes.
Do you have experience with this test?
Look, I think you're referring to immunoassays. They use an antibody to capture the microorganism. Yes. Yeah.
Exactly.
So in my research, I was always interested in using whole microorganism without using any labels or capturing agents. It is also possible. And people in the past tried different things. Even, of course, first you need to capture the microorganism using an antibody, and you need to throw another antibody, which is labeled with a Raman or dye or maybe fluorescence or whatever it's just a basic immunoassay. But the only technique some people use fluorescence, and most people, I mean, there's some, I think, high number of publications showing the surface and Raman scattering can be used. I don't have experience in my laboratory, but I know what it is. I mean, because I know what it is, it's just typical immunoassay. I can help you if you need anything from us, because you need to find the antibodies, okay, for the capture antibody and label an antibody. But I mean, what are you going to achieve with that? Because I mean, I always criticize such an attempts, because there is always a simple technique to identify, for example, just gold nanoparticles. And you can use a typical light microscope to see them if they label antibodies there or not. Of course, sensitivity is not going to be that great, but I don't know. I mean, it depends what you look for, what kind of application you look for. And for example, for bacterial resistance, microbial resistance is a different story.
The good thing about the Staphylococcus that we're working on, and this is why we used it also as a kind of a model organism, is that the resistant protein, the protein that causes the resistance, is on the cell wall. So it's actually on the surface.
Yeah.
So it is easy detectable. You have the gene product on the surface, so you don't have to go into the cell.
Oh, so maybe antibody, if you can make an antibody targeting that Q, molecular Q, which is causing the resistance, maybe you can find out easily.
Okay, okay. My question was actually if you have experience on this, but
no, I don't. I mean, I don't. But I mean, still, that doesn't mean that we can make it. I mean, I know how it works, but I didn't do that in my laboratory.
Thank you. Thank you very much.
Thank you. So, any other questions? And concerns or if not, I think I'll see you around, except Catherine. I mean, who do you work with? I mean, I know.
I'm in the group of Professor Pop, but
Jürgen, yeah.
Yeah, Jürgen, and very closely with Karina Weber and Petra Rösch, and also Ralf Ericht.
I see. Just say hello to Jürgen. So
when I see him, yes, I will say.
Yeah, if you can't see him, I know that. Who knows where he is?
Exactly. I will.
Yeah, and thank you very much for attending, everyone. I appreciate your time and see you around. Thank you.
Thank you very much.